Hello and welcome to Physiological Psychology. Today we'll be talking about the role of the parietal lobe, specifically focusing on our other senses that we haven't discussed yet. So we often discuss, vision and hearing as kind of these main senses, but we know we have many senses, and a lot of them involve our sense of touch, smell, taste. So we're going to be going beyond the parietal lobe, but that's kind of like our starting point in terms of this. And the focus is going to be on the senses themselves. Now, this shows us what we would expect to be reading. Basically, where can we see some supplemental material for this? And this is from Brain Facts and then Anatomy and Physiology from OpenStax, with some suggested areas that are going to be helpful. Most of this just a few pages at a time. So when we talk about these other senses, touch involves things like pain, itch, tickle. You've got different types of pressure. If you push down, you've got deep pressure versus like vibration. If you've ever gotten electric shock, it feels like a vibration because it stimulates those receptors. It can also stimulate your pain receptors too. Our sense of balance, our sense of proprioception, right, telling us where we are in space. That'll be a little more involved when we talk about movement, but it's part of this same kind of touch. Smell, for instance, involves two separate nerves, the olfactory and the trigeminal, which means they're actually getting different types of information that are involved in our sense of smell. Taste really combines 5 different sensations, 5 different types of receptors, and you've got temperature, you've got texture. You might not like something for a certain reason like that. And so we go and we look at, again, our types of receptors. Well, we've spoken about both photoreceptors and mechanoreceptors, but now we're going to focus, because our sense of touch is very much mechanoreceptors. We've also got chemoreceptors to discuss our senses of taste and smell, and thermoreceptors, which is going to be part of our senses too. So perception is the processing and awareness of these stimuli. And remember that we are processing things all the time that are not being perceived, right? So we're picking things up and they're just not reaching to some level of awareness. Our parietal lobe, right, is behind our frontal lobe, so we would say it's posterior to the frontal lobe. Across the central sulcus is our motor area. And one of the things we note that's important to consider is our area of body sensation is near our area of body movement. We talk about movement in the frontal lobe. We're going to be looking very close to this, and we'll be seeing that our movement is right next to our physical body senses, which again should make sense to us. There's a lot of back and forth. Movement involves feedback, lots and lots of feedback, and feedback's coming from our senses. This is also, if you think about it, information coming in to our occipital lobe for vision, and we said that it goes down here to our temporal lobe for our what pathway, and it goes up this way to our parietal lobe for our where pathway. Where is something in space? You hold an item out. Can I reach for it? I'm navigating in that regard. And the parietal lobe is associated with space, navigation, and body senses. Now, you might say, I'm not good at navigation. Perhaps I have some parietal lobe issues. Well, that's more of a thing that has to do with, in a world of GPS and not having to think about it that much, people, I think, don't have to be particularly good at navigation. But if I held out an item and said, hey, grasp this, pencil or pen, you could easily reach out and grab it, right? When we look at somatosensation, these are our body senses, we're referring to the body and its movements, and it includes all of these types of touch: deep, discriminative touch, deep pressure, cold and warmth, our thermoreceptors, right? Pain, itch, and tickle, all separate and somewhat inhibitory. And position and movement of the joints. Now, in this upper image here, we see a somatosensory map. One of the things we see this comes early from the work of Penfield, who tried to kind of label out like what this little homunculus would look like. You've seen these odd little statues with great big hands and faces and tiny bodies. That's what we're looking at here. The idea is how much of your, this cortex is paying attention or processing different aspects of our somatic sensation, and it has to do with really what's the necessity. Clearly, we need to be able to have a good sense of discrimination with our hands. From here, from our lips down through our larynx and pharynx, we have to have extremely dexterous movements, right? Our coordination for speaking. Remember, we speak, we breathe, we swallow, all through the same piping, which means we have had better do a really good job of coordinating those activities. So we see here in this image, quite a bit of it is dedicated towards all the muscles involved in this. Your hand, face, because facial expression is a great part of communication, one would think. If we look at how much is dedicated to these things, we think it must be related. You see how you have your fingers, and you can actually have a musician that fingers a lot, on like strings or a keyboard, can actually have interference here, where they have so much kind of overlap of sensation. Now we see, okay, a little bit of foot, our genitals certainly have sensation here, but we'll see in terms of the mechanics on the movement side, it's not quite as large. And our trunk, our torso, the center of our body, very little sensation. You can test this by using a caliper on the body itself. If you were to put a 2-point discriminator on the palm, something like this, you may be able to say, oh, it's very clearly 2 points. If you use a 2-point discriminator caliper on the back and you said, am I pressing 1 point or 2, you may not be able to tell. There's much less sensation there. But again, we don't need it. Now, for our touch receptors, we're looking at the skin, right? And there's a lot happening in here in this extracellular matrix of the skin. Touch receptors can be a simple bare neuron, a modified dendrite. So you've basically got a dendrite, this bare neuron ending is just the end of a neuron. And so putting any kind of doing it and touching it is going to create some kind of signal. But say you want more information. A Merkel disc, right, is going to be sensitive to other things and is going to be a modified dendrite. You could have an elaborated neuron ending, a bare ending surrounded by non-neural cells that modify its function. Stimulation for these opens a sodium channel. This triggers an action potential. So do you see, for instance, you think of a hair cell, and hair maybe on part of your body. If you were to bend these hairs without touching the skin, right, what you might be doing would be to cause this bending of a receptor. You bend the receptor and it allows these sodium channels to open, which immediately sends a action potential. Let's look at just a few of these and kind of go into depth for what they are. So, the Pacinian. This detects sudden displacement or high-frequency vibrations on the skin. These are onion-like outer structures, and we can see the image here kind of wrapped around, that resists gradual or constant pressure. So what this tells us is it's really tuned for change. Remember, our senses like change. Our vision habituates. We habituate to sound. The same thing for touch. You haven't thought about the fact that you have, a shirt that is draped over your shoulders, right? And it's putting pressure, but this is rarely mildly detected. Sudden or vibrating stimuli bend the membrane and trigger an action potential. Next, we have our chemicals. And so you look at these bare nerve endings, right? These free nerve endings. If you were to cut yourself, you cut through one of these and it sends a pain signal. Chemicals can stimulate receptors for heat and cold. For instance, capsaicin and menthol. can actually cause us to have these experiences, right? These chemicals that make us feel this way. These are things in like the, for pain relief, right? Where you have like those creams and gels. Sometimes they will have like hot patches, like capsaicin patches. Sometimes you will use menthol for this. Now, there's this idea that women are more sensitive to touch, and it's true, but in an interesting way. Basically, these Merkel discs we see here are sensitive to touch, to light touch. The reason women seem to have better sensation here is they're smaller in general, smaller, slimmer fingers compared, to, for instance, compared to you to a male or a female family member. And you look at the relative size of your fingers, that difference is going to be related to how many you have packed in. So you have the same number of discs, but packed in to a smaller area. So someone who has smaller, slimmer fingers, who might be of smaller stature, is simply going to be more sensitive to touch in this regard, assuming the same number of receptors. It's like you don't have the exact same number that I do, but, closer related, more likely. Now, this information from these touch receptors in the head enters the central nervous system through cranial nerves. We call our 12 pairs of cranial nerves, and a number of them are going to be things that we kind of talk about here in terms of taste and smell. So information from below the head come from our 31 pairs of spinal nerves. Now, while the cranial nerves, some of them are mixed, but some are specific to movement or to sensory. Spinal nerves all have both a sensory and a motor component and connect to a limited area of the body. Again, we have more of these up here and fewer down lower. We're also aware that a spinal cord injury higher cuts off more nerves, which is why they tend to be more disruptive. Now here we have our map again, and we know that various aspects of body sensations remain separate all the way to the cortex. This information is being preserved and going where it is needed. And then we have the somatosensory thalamus. Recall the thalamus, center of each hemisphere. These things are dedicated toward kind of like processing, switching. We think of it as like a switchboard or relay station. And these are sent to different areas of the somatosensory cortex. Now there's a story, there's a video you can find called The Man Who Lost His Body, and it's a story of Ian Waterman. This is a fascinating case because Ian Waterman had a severe damage to his proprioceptors. He basically lost his sense of proprioception. Now, when people lose this sense, they no longer are able to move. The muscles all function. They're still sensing things. But you lose this ability to know where your body is in space. Like where, if you're not looking at a body part, you don't see it. You're not aware of it, right? If you were to put, your hand behind your back, you can say, okay, I'm going to hold 3 fingers. Somebody could move your hand and ask you how many fingers you're holding up, and you could feel it. But not so for Ian. Well, Ian was a really, special individual who figured out how to end paralysis, this basic paralysis, and cause movement. So the reason for the paralysis, again, is you have no way of tracking where your body is in space. That's what the proprioceptors do. You're not thinking while you're walking. You're not thinking maybe while you're playing sports. It's all just happening so fast, but your body is keeping track. And we see here these where these things are. These spindles in your muscles, organs, and even your Pacinian corpuscles where giving you information. Ian used his sense of vision. So what he would do is he would be looking at whatever body parts he was using, because when he stopped paying attention, he would lose control. So he had to refocus and kind of attend to whatever it was he was doing in order to maintain control over them. Now we have these mechanoreceptors, right? Muscles, tendons, joints. These are our proprioception organs. we also use vision and our sense of balance. And it's integrated so that you have this single combined sense of where your body is in space. But because of rare cases like Ian Waterman's case, we're able to see that very clearly these combined senses are actually made up of other things like, proprioceptors in your muscles throughout your body. We can also create really interesting illusions, because just like Ian used vision to be aware of where his body was in space, we can use vision to trick you into experiencing pain or fear when there is none present in your body. So this is called the rubber hand illusion. Basically, you'll take a rubber hand, generally something that's long enough so that you can cover it and make it appear. So this person here would have maybe a shirt that comes all the way down here, or this rubber hand would kind of extend. Basically, their real hand, this person's left hand, is being placed behind a screen, so they can't see it. The rubber hand is being placed in front of them. And then each hand, both your hand and the rubber hand, are stroked with a brush. What this does is you're watching the hand being stroked. So here's my hand over here, and then there's a fake hand here, and this fake hand is being stroked with a brush, while my hand is also being stroked with a brush. What this does is it helps to really drive home the illusion that this rubber hand is my hand. Then, and you can try this, you can do this, you would basically take a knife and stab the fake hand, or maybe a fake knife, you don't want to damage it, right? And the person who is undergoing this experience will panic. You can set them up to a GSR measure, heart rate. Their heart rate increases, their skin conductance increases, because they're basically perceiving this hand as their own hand. We know that vision is a primary sense, in the same way that we trust vision over our hearing when we talk about the McGurk effect. When we talk about the rubber hand illusion, once again, we're trusting vision over our sense of touch, over our sense of proprioception. Related to this is also the case of phantom limb. Phantom limb happens when somebody has an amputation. 80% of the time, they will occasionally experience phantom sensations in their missing limb. Some will experience pain, and this is likely due to reorganization of the somatosensory area. Look at, for instance, how much of the somatosensory area is dedicated to a hand, right? Well, if you were to lose your hand below the elbow, you have a large number of neurons that are now not getting any input. Well, these neurons aren't just going to say, oh, well, I guess we're not doing anything now. They're going to go do something else, but they're still going to give you the sensation of that hand. Now, what can happen is people who have an amputation can sometimes have pain because you don't get the feedback. So, for instance, you would have this signal that's saying, clench your hand, clench your hand, clench your hand, right? But under normal circumstances, you would also get this feedback, be like, ow, that's too much. That's too much. But if you have no hand, there's no way to give feedback and say, hey, that's too much. So you've got this clench signal, this experience of clenching without the feedback. Well, Ramachandran, a researcher at the University of California, San Diego, created what's called the mirror box, and basically created this almost a phantom limb amputation experience, right? So what they do is, in this mirror box, you would have the person put both of their, hands, one actual hand and one phantom arm, into the box. Now they're only looking at one part of it that has the reflection. So it appears from their perspective that they now have two hands. They would clench as hard as they could and then release. Now this hand is not there, so they would clench, they would see this hand in the mirror and then release. And what would happen is they would actually experience relief. Because again, we're giving this visual feedback, which tells us we are getting a lot of feedback, sensory feedback, that we are simply not attending to and not regularly aware of. Pain is caused by something that is harmful, and it's not necessarily a bad thing, right? The point of pain is to draw our attention, right? Something's wrong. You ever been outside barefoot and all of a sudden you feel a pinch, right? And you're like, ow, and you step back. Well, maybe like one of those fire ants or something bit you, and it hurts, but it's like, hey, something's wrong. Get your foot out of here. And there you go. You know, less damage happens, right? These are these bare nerve endings cutting in, causing pain. Now, some pain receptors also respond to things like acids, heat, or cold. You have capsaicin, we mentioned, capsaicin patches. So with severe pain, you might take a heat patch, put it in the area, and what happens is it creates this block, all right, because you can only get so much information. We'll kind of look at how this works, right? So axons carrying pain are generally unmyelinated, which means it travels slowly. I mean, it's still traveling quickly, but slow compared to other information, which means you can be getting the information that something's wrong before you feel pain. If you've ever burned yourself, if you ever grab something that's hot and you pull your hand away, you know it's going to hurt, right? You're like, oh no, I just burned myself. Before you even feel the pain, you know that you've burned yourself. So because you have this sense of, oh, that was really hot, oh no, I burned myself, maybe you can start, treating the pain before it gets worse. Mild pain triggers the release of glutamate in the spinal cord, and stronger pain triggers the release of something called substance P. This is related to the increased sensitivity, the intensity of pain. Pain has both the physical and the psychological components, and we increase the psychological. So, for instance, the somatosensory cortex responds to these painful stimuli, memories of pain, and signals that warn of impending pain, right? We also have areas like the thalamus, amygdala, hippocampus, prefrontal cortex, and cingulate that are associated with emotional associations to pain. Which means, again, we have areas where if you see somebody experiencing pain, you can have some of these areas active. The memory of pain, okay? So it's not just the sense of it, it's actually these other things involved too. Opiates work, right? Drugs that are given for pain, like morphine, codeine, Percocet. These block the release of substance P, which minimizes the experience of pain. And we also they mimic endorphins. And morphine comes from, the word endorphin comes from the word for endogenous morphine. Because first they discovered morphine, and then they discovered, oh, we've got these chemicals in the brain that do the same thing. And so that's what they do. They attach the same brain receptors as morphine. Endorphins are things involved in something like runner's high, right, where you're exercising and you just feel really, really good. Big burst of endorphins. Things that are pleasurable will often give us a release of endorphins. One of the reasons that opiates are so wildly addictive is endorphins are wonderful, right? They feel really good, and if you have them all the time, you want them all the time in that regard. Now, the gate theory, if we look at how we can kind of block pain, right? How does the capsaicin patch work? The idea here is you've only got so many nerves to get information. So as you're kind of getting these receptors, there's these other areas that can provide info that can close the pain gates. A capsaicin patch, right? Let's say you have pain in your leg. You take that patch and you put this patch. Now, generally the pain is going to be a small area of pain, but the patch is a large area that causes a warm sensation. So even though pain is a very sharp, unpleasant experience, and this heat patch is very mild warmth, you're getting a lot of warm signal, which is actually interfering with the pain signal. So you decrease the pain perception. You can also oftentimes decrease the pain perception by looking at the area that's in pain, right? So it kind of does this. Sweet smells and tastes can help. Certainly distraction can help too. And we also see this example of the emotional response where placebo, right? A placebo is a drug that doesn't do anything, but if you believe it works, it can have an effect. So here we see, someone says, here's a pill, it's going to reduce your pain. You may actually feel reduced pain. It decreases pain perception by decreasing the emotional response to the pain, not the sensation itself, but you think that it's going to be less, so it is. We see that this is happening in the cingulate cortex, but not the somatosensory. The other side of the placebo is called a nocebo. A nocebo is something that someone tells you, take this, it's going to cause you pain. And then people will experience it. One of the things that's changed over time is they don't tell you the side effects as much as they used to of medications. Because if you're expecting it to happen, it's more likely to happen. Now, emotional pain resembles physical pain in many regards. And there's this experiment that they do where they have somebody experience exclusion. So let's say you found out that, all of your best friends, all the people that you know well and hang out with had a party and they didn't invite you, right? That sucks. That feels bad, right? Well, one of the things that happens is we see this activity in the cingulate. This is emotionally painful. And they found out that Tylenol, right, acetaminophen in the United States, it's reportedly decreases the amount of emotional pain. So Tylenol is one of those interesting drugs given for pain that not only reduces your actual pain, but it reduces your experience of pain. Now, Tylenol is a dangerous drug because you take too much of it, it can cause toxicity, right? But in small doses, when in intense pain, it works really well. In fact, there's a lot of support for the notion that drugs that we take over the counter for pain work as well as opiates without these addictive problems. The problem is they are much more toxic to the body, where opiates aren't, but they're a side effect of addiction. Now, itch is not the same thing. So you ever you scratch that itch, what you're causing is pain, and pain inhibits the itch, right? Histamines produce this itching sensation, and this is in the skin. In the brain, histamines keep us awake. So it's interesting how these things work in different areas. And they activate this distinct path in the spinal cord from the skin. Impulses travel slowly, comparably. Now, 0.5 meter per second isn't slow when you consider, 2 meters tall? Okay, so it takes a second for that to travel. But we see this inhibitory relationship where opiates, for instance, increase the experience of itch, and histamines decrease itch. When you scratch that itch, you cause pain, which inhibits the itch. Two separate senses. So now we go from our these physical senses to our chemical senses. And chemical coding is along these lines here. So you've got these two theories and two ways that this works. In mammals, we have a cross fiber pattern. And in other species, you have this labeled line species, generally invertebrates. Labeled line means a single kind of receptor is going to give you a signal like, oh, there's something present in the environment. Cross fiber means each receptor responds to a number of stimuli, a range of stimuli, and they contribute to the perception of each of these things. our taste sense refers to the stimulation of the taste buds. These are receptors on the tongue. And what we're looking at here is the endopiriform cortex, right, which is inside kind of the piriform cortex. So this is the piriform, endo would be further in. And our perception of flavor is the combination of both taste and smell, right? Maybe if you have a cold, things don't taste as good because your nose is maybe plugged and you're not able to kind of get that scent information for flavor. Okay. Now, we look at where these receptors are. Again, here we're looking at the area around the hippocampus, right? We're in the temporal lobe here. And we're really kind of related between the temporal lobe and the insula. So here's our receptors for taste. And these receptors are modified skin cells that are replaced every 10 to 14 days. You ever burn your mouth on some food, maybe a hot beverage, or why is it always the cheese on the pizza, right? Well, they're going to start replacing themselves, and they're replaced every 10 to 14 days. So your sense of taste comes back fairly quickly. What we have here are papillae. These are the little structures on the surface of the tongue, and here you see kind of like the, where these are on the tongue and kind of what they look like. You've got the taste pore and the taste cells, and these are going to connect to your nerves that are going to send information on taste. Each papillae may contain up to 10 or more taste buds. Each taste bud contains about 50 receptors, and most of these are located along the outside edge of the tongue. There is a weird myth, and you might have seen this growing up, where you know your primary tastes, and you'd see a little map of the tongue, and on the tongue you would have, here's where bitter is, here's where sour is, here's where salty is. Nonsense. Utter nonsense. They're spread out along the areas of taste in the tongue. Now, you can alter one receptor but not others to identify taste receptors. You can get some chemicals that are harmless chemicals that can knock out certain receptors and temporarily and change the way things taste. Adaptation refers to the reduced perception of a stimuli due to the fatigue of receptors in the same way everything else is. You can fatigue your taste receptors if you, for instance, start eating salty foods. In the beginning they taste salty and then they're not so salty. And because you've kind of adapted to the taste. Cross-adaptation refers to reduced response to one stimuli after exposure to another. Now here's our 5 primary flavors: sweet, sour, salty, bitter, and umami. The salty receptor permits sodium ions across the membrane. So that makes sense, right? What causes your nerves to fire? Sodium ions. So you have a taste for salt. You basically have a receptor that just says, okay, salt's present, just let it in, and boom, here's a signal. Sourness closes potassium channels. Acid binds to the receptors, and this depolarizes the membrane for information on sourness. So salty and sour are rapid. Okay, these are ionotropic. Sweetness, bitterness, and umami are all metabotropic because they activate a G protein that releases a second messenger in the cell when a molecule binds to a receptor. A slower process. Again, still so rapid it's almost instantaneous for us, but orders of magnitude slower for this because it does take time for this second messenger to send the signal. Now, bitter receptors are sensitive to a wide range of chemicals. We have about 25 types, and they generally send information about toxicity. Things that are bitter, for instance, animals that eat foods that we would think are bitter tend to have massive livers, and they spend a lot of time digesting these things because you have to filter out these kind of toxins. So bitter generally is a signal that says don't eat that. But again, we have a taste for bitter things. Sometimes we get used to these things. And most taste cells contain only a small number of these receptors. We're sensitive to a range of harmful stimuli. Some people, now again, this is one of those interesting things with genes. If you remember the PTC example, right, where we experience mild bitter, we might not experience bitter, we might experience intense bitter. You want mild bitter to a lot of things rather than intense bitter to a few, and then having no sensitization for bitter for other things. Taste nerves project to a structure in the medulla known as the nucleus of the tractus solitarius. This projects information to various parts of the brain. And with taste, we have cranial nerve 7, the facial nerve, 9, glossopharyngeal nerve, and 10, the vagus nerve. All of these things are contributing to taste. And we look at kind of where these things are. Basically, we're looking at deep in the medulla pons region for where these things are connecting. So 7, 9, and 10. And we're pretty well aware of the vagus nerve because that's the wandering one that runs all the way down into our intestines. But 7 facial and 9 glossopharyngeal, we don't tend to think of that much. More specialized. Now, taste, we've got various areas of the brain responsible for processing this, but heavily we've got the insula, the primary taste cortex, which is behind the temporal lobe. Somatosensory cortex responds to the touch aspect of taste, right? Is there something you don't like because of the way it feels? You don't like the way that sensation, but the taste isn't there. Mushrooms don't have much of a flavor on their own, but they can have an odd texture that people don't like. Now, each hemisphere of the cortex is also responsible to the ipsilateral side of the tongue. So you're getting information from both contralateral and ipsilateral. There's also, we see like the temperature issue with taste too. Now, not everyone has the same sensitivity to taste. You can actually count your papillae to see how sensitive you are by looking at an area of your tongue. And you have some people that are called super tasters, highly sensitive to flavor. These people might be more likely to be foodies, but anybody can, kind of develop their sense of what they enjoy in terms of the complexity of tastes. With our sense of smell, called olfaction, we are detecting and recognizing chemicals. Now, chemicals, most of these things we're talking about are organic chemicals. They're telling us about food and danger. Humans can follow scent trails. What they do is they will blindfold somebody and they'll have like a trail of scent, right? And you can see dogs. You ever see a dog kind of like put his face down and start sniffing along the ground following a scent trail? Humans can do that too. It takes practice, but we can learn to kind of attend to this sense. It's also important for our food selection. It's linked to our sense of taste. Now, we have some interesting sensitivities too, where we tend to prefer the smell of potential romantic partners who smell different from ourselves and family members. This decreases the risk of inbreeding and increases the probability humans that our children will have a wide range of immunities, right? So we look at the dangers of inbreeding, but we also see really the benefits of not, right? You want someone who is different in terms of the immune system genes. And it gets even more interesting when it comes to the things we're sensitive to. Our olfactory cells, these are neurons that line the olfactory epithelium. So basically, here's your sinuses. This is all in here, right? Back through in here is like kind of this spongy sinus material. Much you can break this much more easily than skull. And up inside your sinus patches, you have your olfactory epithelium. Receptors are located on cilia, which are these tiny little hairs which extend from the cell body into the mucous surface of the nasal passage. And these, we have hundreds of olfactory receptors. We're sensitive to many things. Well, proteins in olfactory receptors respond to chemicals outside the cells and trigger changes in G protein inside the cell. And again, whenever you see G protein, you think second messenger, you think metabotropic. It's slow. The only things that we saw fast were like the sour and the salty. The G protein then triggers chemical activities that leads to action potentials. Axons from olfactory receptors carry information to the olfactory bulb. And we see the olfactory bulb is right there, right by our memory, our emotion centers, by our thalamus. Similar chemicals excite neighboring areas. Receptors regenerate in about a month, but they can be permanently destroyed. And we've got individual differences, with women tending to have stronger neural responses to odor. The ability to detect a faint odor and become more sensitive to it is characteristic of young women, young adult women, I should say. It seems to be influenced by hormones, and it should make sense that hormones change our sensitivity to things over time. Mice that have a gene that controls a channel through which most potassium travels to reach the olfactory bulb develop a sense of super smell. Now, we also consider the last air we talk about smell is pheromones. The vomeronasal organ, the VNO, is present in a number of species. We see it here in snakes. It's also called Jacobson's organ. Basically, this is what is part of our olfactory process that's sensitive to pheromones. Pheromones are chemicals that they don't have a smell, but they affect us. So, for instance, if you ever hear dogs like going crazy, right? Oftentimes, and there's no reason for it, like the dog wants to get out, it's bark, bark, barking, there's nothing there. There could be, if it's a male dog, there could be a female dog that is, basically producing pheromones because she is ovulating, and that dog is getting the signal that says, I need to mate now. Now, whether these things affect humans is much more interesting. It does seem that there is the case, right? So it affects us. An animal can change the effect, the behavior of another animal. Again, this is for the process of, reproduction generally. But with humans, our VNO and pheromones are really debatable. We know that it does affect. For instance, women who live together will often have their menstrual cycles synchronized. This happens through pheromones. It's also possible that if somebody ever looks different to you, right? Like all of a sudden you're like, wow, this, I don't know why this person appears so attractive today. It could also be due to pheromones, right? Humans, it's tiny, has almost no receptors. For a while it was believed that humans didn't even have these. However, we do certainly show that we unconsciously respond to some pheromones through receptors in olfactory mucosa. So there is some effect of pheromones. The issue is it's unclear how much and how powerful this effect is. But whatever's affecting your behavior without you being aware of it, that's kind of terrifying, right? We like to think that, no, I'm a human, I've got, sense, and I can consider these things. But really, we know that to a certain extent. That is the end of our segment on our senses, going through, our somatosensory, sense of touch and all the parts of the sense of touch in our parietal lobe. And we've also talked about our taste and smell, our insula and our associated areas there. Thank you, and goodbye.